Welcome back, students. This is Module 4.2.4, Cloning. You'll recall that last time we were telling you about some of the techniques used by biotechnology. We told you about selective breeding or artificial selection, in which humans select for desired traits and control them by controlling the parentage in plant or animal breeding. A second type was recombinant DNA, in which restriction enzymes were used. We talked about that earlier as well in a previous module. And the third technique we mentioned last time was DNA fingerprinting. This time we're going to talk about cloning. And cloning is defined as making a copy or a genetic twin of an organism. And there's two types of cloning: reproductive cloning to make a new offspring, and therapeutic cloning. And we'll talk about these. Therapeutic cloning uses stem cells. Now, natural reproductive cloning is fairly common among many plant and animal species, probably more plants, but there are a number of animals that also reproduce in this manner. Think of the aspen trees up on the Huachuca Mountains. You can see individual patches of yellow. And the ones that turn yellow at the same time, the leaves change at the same time, are genetic clones. And there are many other examples. Bermuda grass and strawberries also clone as plants. Let's talk about artificial reproductive cloning, though, rather than natural reproductive cloning. Artificial reproductive cloning is what biotechnologists do. The first vertebrate animal with a backbone that was cloned was Dolly the sheep, who was cloned back in 1997. And there was a Scottish scientist named Ian Wilmot who accomplished this task. What he did was he took a diploid cell from the teat, or from the mammary of a lamb, and he put it into an egg that had the nucleus removed. We call it an enucleated egg. Remember that an egg is haploid, but he put a diploid nucleus into it. And the hope was that technique, in using that technique, was that the egg with the diploid nucleus would behave as a zygote, a fertilized egg. And to induce it to behave that way, they used an electrical shock. This helped the nucleus to enter into the cell, but it also was supposed to start the cell with the new diploid nucleus dividing, just as a zygote would. And so the embryos did start to divide, and they were placed in the womb of an adult female sheep, so in the uterus of an adult female sheep. And of all those trials, they tried many hundreds. As a matter of fact, only one out of 277 trials succeeded. And since that time, scientists have gotten a little bit better at the techniques, and they've cloned mice and cattle and goats and pigs and even more. But not without problems. Let's look at a kind of a cartoon view of what happened. So we can see right down here that the egg cell was haploid, but it didn't matter because the egg had everything it needs to form a new zygote, and the nucleus that has the chromosomes, a haploid number of chromosomes, was removed. And then they took a cell from the udder, from the mammary gland of another sheep, and it doesn't matter. They could have taken it from the skin or the liver or whatever, but they took it from the udder, and it has a diploid nucleus. And they took that nucleus, the diploid nucleus, and they placed it into the enucleated egg. They gave it a shock to start it dividing, and there we go. Hello, Dolly. As we said, it wasn't very successful every time or anything like that. There were problems. This is showing a little micro technique in which they're using a cytological technique. This thing over here has a vacuum that holds the egg in place, and this they're actually trying to physically manipulate it. It's kind of interesting. Now, cloning, although quite marvelous, it's not without problems. And so reproductive cloning, one problem is that most clones die in late pregnancy, not always for reasons that are understood. If the clones are born, that's often typical that they will die soon after. And many of reproductive clones develop something called large offspring syndrome, in which they tend to be oversized relative to others of their species. Now, this last one, it says development into adults goes haywire. That's a technical term. In fact, it's not. The technical term is genomic imprinting, and somehow it's lacking in clones. Genomic imprinting is when during the developmental process, certain of the genes are turned on or off. And this doesn't happen correctly in reproductive clones, and that's one of the reasons that scientists need to work with them more to understand this process better. Now, we also have to consider who would want a reproductive clone, and what currently reproductive cloning is illegal in the United States, but just think who would want a genetic twin. I mean, when two parents have an offspring, it's genetically 50% like the father and 50% like the mother. If you have a reproductive clone, it'll be 100% like the parent that donates the diploid cell that's put into the enucleated egg. And so it'd be kind of like having a twin that's decades younger in age. And so it'd be kind of odd. Anyway, there may be psychological issues, I'm not sure. But nevertheless, it is illegal, and no one is advocating to legalize reproductive cloning, really. It is more therapeutic cloning where scientists see promise and think that it should be allowed. In therapeutic cloning, this involves producing an embryo that's genetically identical to a particular donor. Usually the donor will be sick, have some condition that they need stem cells for. And the whole purpose for producing a embryo in this situation is to provide a therapy for the sick person, hence the name therapeutic cloning. The clone is only used to produce stem cells. The clone is typically, at least it has been, typically destroyed in harvesting those stem cells to serve as a therapy for the person who donated the cell that created the embryo. And so there's no intent that that embryo should ever become a human being or anything like that. But so you take a diploid cell from the patient and you place it into an enucleated egg, and in about 5 days or so, the embryo will be developed enough to harvest stem cells, and at this stage it's called a blastula or a blastocyst. And those cells that are harvested are called embryonic stem cells. They're called stem cells because they have the ability to give rise to many other types of cells. And so all of your cells, all the other cells in an adult human body are derived from stem cells during the embryonic process, embryonic development. Now, the reason that you have to use a nucleus from the patient is because the cells are going to be re-injected into that patient. And if you take cells from a different person and inject them in, there will be an immune response and the body will reject them. And so to have an immunocompatible stem cell transplant, you need to have the cells from the original patient. And so it's all about avoiding this immune rejection. Here, this actually shows a blastocyst, just a few cells. And you can see that they're removing that, and it will actually destroy the embryo. And some people find this, and this will be one of the turning points upon which our debates will hinge when we have stem cells or therapeutic cloning as a debate topic, which we don't infrequently do. There is, however, it's not like therapeutic cloning is all worked out and working perfectly. There are really virtually no therapies that are approved by the FDA using embryonic stem cells at this time, and that's because there are, as I said, a few problems that have not been solved. the cells may not divide properly. There have been instances in which cancer can develop from the use of these cells, which aren't fully understood now. But it doesn't mean that necessarily that the best course of action is to stop and say what can lead to cancer. But research continues in the hope that we can understand this, because it seems to be a technique with incredible potential to help people. And so this kind of gives us a cartoon overview. There's our patient. They take a diploid cell there, and they take an egg, an oocyte, which is an ovum or an egg, and they put the nucleus in a nuclear transfer, and then you get a zygote that is genetically identical to the patient, and they grow this in vitro, or in a culture in glassware, and then the hope is that those stem cells could be harvested and used to treat a variety of different cells. Of course, they wouldn't use all of them. They would, they wouldn't use stem cells because most likely this patient doesn't have problems with all these different systems. But cardiomyocytes, for example, this person had had a heart attack and their heart muscle wasn't robust enough to pump blood to their body, then they could inject these embryonic stem cells into the heart, and the heart would become healthier and able to pump more blood. Anyway, that's the hope. As I say, these are not approved. These therapies are not approved and being used now. But see down here, the goal is an immunologically compatible transplant. So let's talk a little bit more about stem cells. So embryonic stem cells are cells that have the ability to form any body tissue, and we have some technical terms that are used to describe that. The term totipotent is one term. Tota means totally potent, or totally able to give rise to all the cells. Another term that's sometimes used to describe embryonic stem cells is pluripotent, plura as in plural, as in having the ability to give rise to many cells. And I just note here, I think I already said this, the stem cells are formed 5 to 6 days after fertilization, and they're found in the structure called a blastocyst. And we can see, of course, that some of the potential stem cells can possibly be used to fix damaged muscle or nerve tissue, both types of tissue which divide very slowly. And so this is something that is hope that can be accomplished. They have done such things with mice where they have actually broken the back of a mouse, and it has been paraplegic. Its hind legs won't work, and they actually have injected these, and then the nerve cells in the spinal cord have regrown. They're actually able to use their legs to some extent, not necessarily exactly the same as before they were paralyzed, but similar. And so here's actually some human embryonic stem cells. Here, this shows the promise of stem cell research. So cultured pluripotent stem cells, and then we can get tissues or cells for transplantation and do all different things. It says we could identify drug targets and test potential therapies. We can use the stem cells for toxicity testing. We can use them to transplant to bone marrow for leukemia or chemotherapy. You don't have to have a bone marrow donor, but you can produce your own bone marrow cells from stem cells. Nerve cells that could serve to replace cells damaged in certain devastating neurological conditions such as Parkinson's or Alzheimer's diseases. These are devastating diseases right now that run their course. But we could possibly intervene and have a better outcome for such patients. Heart muscle cells for heart disease, so if you've had a heart attack. Pancreatic islet cells for diabetes. It's the islets of Langerhans. That's the name of the cells that produce insulin. We could also learn to study cell differentiation, and so we could understand and prevent birth defects. And of course, we don't know what else we could discover, and that's the thing with science, we're never sure. So there's a lot of potential benefits there. There are also many challenges. We remain in the basic research phase, but certainly there's a lot of things here. I'm not going to go through every point on this slide, but you can look at it and see all the things that we don't understand well, that we need greater information in order to be able to fully realize the potential, the immense potential of stem cells. But we're in the very beginning stages, and we don't know, again, how we're going to solve these problems, but it will only be through continued research that they will be solved. Adult stem cells. Some people who find the use of embryonic stem cells ethically objectionable because of the destruction of an embryo, because remember, to get those cells, we have to kill the embryo. Actually, there have been some scientists in, I believe, a lab in Korea has worked out a technique to harvest stem cells and not kill the embryo. But it doesn't really matter, because the patient doesn't want an embryo. They don't want a baby. But they just did this to appease those that find this ethically objectionable. In any case, an alternative to embryonic stem cells is adult stem cells. And these are, as it says on the slide, tissue-specific stem cells. So in muscle cells, we have particular stem cells that give rise to other muscle cells. They tend to be further differentiated than embryonic stem cells. So embryonic stem cells can give rise to any tissue, but adult stem cells have already differentiated a little bit, but they can still give rise to more of the type of tissue from which we find them. And so this seems to have great potential. And there actually is at least one therapy, I believe, approved by the FDA, the use of adult stem cells. Again, we can use stem cells from the nervous system to treat MS, which is multiple sclerosis, in mice. Blood stem cells have been used in humans to replenish bone marrow of cancer patients after they had radiation treatments that destroyed their marrow. But there is a problem with adult stem cells, and that is if we weren't even aware that certain types of tissues had adult stem cells until fairly recently, and that is because they are very hard to find at times. And so we can have a severe problem finding the tissue-specific stem cell that is needed. And of course, their partial differentiation limits them to only the type of tissue from which they have been harvested. And so this is just a little bit of information on cloning and stem cells. I'm sure we'll hear much more in debates on the topics. And this will conclude this module.